Photosynthesis is the fundamental process by which plants produce glucose, a vital source of energy and a building block for growth. This process takes place in the chloroplasts of plant cells, where chlorophyll, the green pigment, captures light energy from the sun. Photosynthesis involves converting carbon dioxide from the air and water from the soil into glucose and oxygen, using the absorbed light energy. The overall reaction can be summarized by the equation shown on the screen. The glucose produced during photosynthesis serves as an essential energy source for the plant, driving various metabolic processes. Plants use the glucose produced through photosynthesis in several key ways. Firstly, glucose is used for respiration, a process that releases energy needed for growth and cellular functions. During respiration, glucose is broken down into carbon dioxide and water, releasing energy stored in the glucose molecules. Secondly, glucose is converted into cellulose, a structural carbohydrate that strengthens the cell walls and gives the plant its rigidity. This is crucial for maintaining the plant's structure, allowing it to grow upright and withstand environmental forces. Thirdly, glucose can be stored as starch, an energy reserve that can be broken down and used when photosynthesis is not occurring, such as during the night or in winter. Fourthly, glucose is used to synthesize amino acids, which are the building blocks of proteins. These proteins are necessary for the growth and repair of plant tissues. Lastly, glucose is involved in the production of lipids, which are stored in seeds and serve as long-term energy reserves for the plant, particularly during germination. The rate of photosynthesis is influenced by several limiting factors, which are conditions that can restrict the process if they are not present in adequate amounts. The three main limiting factors are light intensity, carbon dioxide concentration, and temperature. Light intensity is crucial because photosynthesis relies on light energy to drive the chemical reactions that produce glucose. When light levels are low, the rate of photosynthesis decreases because there is less energy available to convert carbon dioxide and water into glucose. As light intensity increases, the rate of photosynthesis also increases, but only up to a certain point. Once all the chlorophyll molecules are fully engaged in the process, further increases in light intensity will not boost the rate any further. Carbon dioxide concentration is another critical factor because it is one of the raw materials needed for photosynthesis. If the level of carbon dioxide in the air is low, the rate of photosynthesis will be limited, as there is less carbon available to form glucose. Like light intensity, increasing carbon dioxide concentration can increase the rate of photosynthesis, but only up to a certain level, after which the rate will plateau. Temperature also affects the rate of photosynthesis because it influences the enzymes that drive the chemical reactions involved in the process. Photosynthesis typically increases with temperature, as the enzymes become more active, but only up to an optimal point. If the temperature becomes too high, the enzymes can denature, meaning they lose their structure and functionality, causing the rate of photosynthesis to decline sharply. Conversely, if the temperature is too low, the enzymes work more slowly, and the rate of photosynthesis decreases. Understanding these limiting factors is essential for optimizing plant growth in agriculture and horticulture. By controlling light, carbon dioxide levels, and temperature, growers can enhance the efficiency of photosynthesis, leading to healthier plants and higher yields. In natural environments, these factors often fluctuate, causing variations in photosynthetic rates and influencing plant productivity and survival. The ability to manage these conditions effectively allows for the maximization of photosynthetic activity, contributing to the overall health and growth of plants.